So welcome to our first asynchronous video. This is going to cover the scientific method. And our learning objectives for this video are to describe the steps of the scientific method, to define some key terms like hypothesis and null hypothesis, as well as independent and dependent variable, and then to end by designing a well-controlled experiment. So to do that, we're going to talk about the steps of the method, talk about our definitions, and talk about experimental design. First, I want you to think about what is the scientific method and what are the steps involved. And as biology majors or science majors, this is something that you should be pretty familiar with at this point in your education. And so I want you to pause the video and go to this link that's posted in the agenda for a Google Doc entitled Class Notes Week 1 Scientific Method. And there are instructions on that Google Doc about what I want you to do. But basically, you're going to brainstorm with your classmates about the scientific method. So pause the video, go to that link, and I'll wait right here. Okay, I hope you did that. Let's keep going. Let's see if you were right. Let's see how good you guys were able to be in coming up with the steps of the scientific method. Okay, so the scientific method is a protocol to develop a tentative solution to a question or a problem, to gather information, gain knowledge, evaluate that known information, analyze data, form conclusions, and end by sharing our findings. And it's important because it's used by scientists across the board. So everyone from the microbiologists who study the littlest things to the astronomers studying the stars. We all use the scientific method. And it's got a few basic steps. So it involves an observation, a question, a hypothesis, an experiment, an analysis, a conclusion, a report, and then the sharing of that report. And we'll dive into each of these steps in particular. It's important to keep in mind, however, that science is ever changing and it's always open to new information. So as you move through this process, it's not that you reach the conclusion and you just end and it's over. It's that the conclusion will then inform and go back to the hypothesis and maybe have you refine your hypothesis and test it again in a new experiment. Or it could lead you to a new question that would come up with a new hypothesis. And so it's a process that keeps going and going and going, as opposed to something that has a definitive endpoint. And because of that, the ideas that we have about science is also changing. So what the theories are and what's considered known is constantly in flux as we gain more information, as we go through this process across a scientific community and come up with our own conclusions. And so let's dive at the beginning here. So a question. These mainly come from curiosity about an observation. So you observe something, you notice something, and that leads you to come up with a question. Now, a good question is something that is testable. So it has to have some sort of experiment that will come from that. You can't just say, I wonder what the best state is, right? That's not a good question. There's no way to test best. Right? But if you instead said, I want to know what the biggest state is, well, that involves something that you can measure. It also wants to be something that you can repeat. So you don't want to study something or ask a question about a one-time event. You want to ask a question that you can repeat and test over and over again. A good question is controllable. A good question has an ability to just alter one variable as opposed to multiple variables. And it's novel in some way. And when I say novel, I don't just mean that the only good studies are studies that have never been done before. There is something called a replication study. And replication studies are just that. They're when you repeat something that someone else has done. But replication studies are important, but they don't always make for the most interesting studies. So a good question is something that is new. A good question is something that is new. So again, we're looking for things that are testable or measurable, things that are repeatable, things that are controllable, and things that are novel in some way. And again, the novel part is probably the least important of those characteristics. So after you come up with your question, you then want to do a literature review or a lit review. You want to figure out what is known about that question already before I even dive into it. And from there, you're going to come up with your dependent variable. This is the thing that you are measuring, the thing that's changing, and that depends on your independent variable, which is the one that you manipulate. So you're going to change and alter your independent variable and then measure the effect it has on your dependent variable. So if I was interested in the effect of salinity or how salty water is on plant growth, I would change the salinity of water. That would be my independent variable. And I would measure the growth of my plant. That would be my dependent variable. And the way that you can think about this is to think about the graph that you would get from those results. The independent variable is going to go on the x-axis, and it's going to be, again, that thing that you're manipulating or changing. And the dependent variable is what you're measuring, and that's on the y-axis. Okay, so this leads us to our hypothesis. And a hypothesis is not a prediction. So you're not trying to see into the future. Instead, you are offering an educated explanation of how that independent variable affects our dependent variable based on the previous research and data that you've studied and come from your literature review. And a well-formatted hypothesis has 4 components. It's got an independent variable, a dependent variable, a species, and a biological reason. So you want to say something like, if independent variable affects dependent variable because of some reason, then the manipulation of that independent variable should have a predicted effect on the dependent variable in species. So if salinity of water affects plant growth because of, I'm not a botanist, but some sort of botany reason that you got from your literature review, then increasing the salinity should have some sort of predicted effect, should increase the growth of tomato plants. Okay, so you want to follow that format. And there are 2 types. The null hypothesis predicts that there's no effect. So for this, it would be the null hypothesis would be that salinity, increasing salinity, has no effect on plant growth, right? There is no effect of that independent variable on our dependent variable. The alternative hypothesis, on the other hand, predicts that an effect will happen. So the null hypothesis says salinity will not affect plant growth, whereas the alternative hypothesis says salinity will affect plant growth here for our tomato plants. An important caveat is that a hypothesis cannot be proven, right? It can only be supported or rejected by data. So it's not that you prove 100% without a little bit of doubt that this is true or not true. You instead say, the data I have collected supports this hypothesis, or the data I've collected rejects this hypothesis, okay? You're instead building your case as opposed to saying black or white, 1 + 1 is 2. This is not math. We don't deal in indefinitives. Okay, this is going to lead us to our experiment, and there are 2 types of experiments. There's an experiment, and there's an observational study. An observational study is the kind where you're just observing what's happening without making any alterations. So if I thought, again, salinity affected plant growth, rather than adding water with different salt concentrations, I could just look at the plants that are growing, measure their growth as they're growing, measure the salt in the water that is given to them without controlling for anything. Whereas an experiment, I'm going to alter that independent variable myself, and I'm going to measure my dependent variable. For an experiment, you need to have certain groups as well. So you need to have your 2 control groups. The negative control group is the one that you know will have no effect. So you could just use plain water. Plain water is our baseline. The positive control is not always available to you, but if you know a certain thing really helps, that could be your positive control. So fertilizer, for example, mixed into water would be our positive control. We know that that's going to have a positive effect on plant growth. And then your experimental groups, so the different salt concentrations here that we're testing. Our experiment needs to be controlled. So we don't want to have different amounts of sunlight, different soil components, right? We only want to be changing that one variable as opposed to everything else changing. Data collection, we need to be accurate and detailed in our recording, keep notes, record all possible data within reason, and keep it organized in a notebook or a series of electronic data sheets. So let's do an experiment here. So our hypothesis testing, we're going to incorporate controls to ensure that the effect being measured is really the result of the variable being tested. So if our hypothesis is that the application of 20 milligrams of tea extract will increase the lifespan following a heat shock in C. elegans, and this is the kind of research that I do in my research lab. So to do that, we're going to take 3 different groups of worms. This is me attempting to draw a worm on PowerPoint. That's why I labeled them. So you know that these are nematodes, these are C. elegans. So we're going to have our experimental group. They're going to be exposed to the 20 milligrams of tea extract. We're going to have a negative control, and this should have a non-response, right? So this is just the water without any tea. And then we're going to have positive control, which should have a really good response, and it comes from our literature review. So for this, we're going to use resveratrol, which is an anti-aging compound that's found in red wine and is known to have a positive effect in nematodes. Okay, so after we heat shock them, we expose them to the different conditions, the tea extract, water, resveratrol. We measure survival. The water group all dies. That's our negative control. Positive control, they all live. And then for this experiment, maybe we found that it had no positive effect. They all died. The response here is no increase in lifespan. And although it seems illogical, a non-response is something happening. It's still measurable. Okay. Once we do that, we're going to analyze our data. We're going to calculate means and variance, and then we can plot our data into graphs to look at trends, differences, and outliers, and run statistics. And all of that we will talk about and get to in this class. And then finally, we want to interpret, explain, compare, and conclude. So you want to say, how did our independent variable affect our dependent variable? What does it mean? Why do you think that this is happening? Did it support your hypothesis? If so, great. But if not, that's fine too. Your hypothesis doesn't have to always be correct. In fact, if your hypothesis is always correct, people are going to start to think something's fishy. So it's okay if it's not right. There's no such thing as bad data. So the data is the data. It's not good or bad unless the researcher made some sort of big mistake, okay? The data is the data. There might have been issues with the experiment. There might have been variables that were not well controlled. But all of that you can learn from. And you want to try to be as objective as possible. So you don't want to get emotionally invested in your experiment and the outcome. You want to compare your findings to the literature. Do they agree? If so, do they fill any gaps in the knowledge that existed at that time? Do they contrast with what you found in the literature? Why do you think that is? What are some differences you could find? And then what are the takeaways? Did you answer your question? What's next? Did you fail to answer your question? What could you change to solve that problem? And then finally, you want to share your findings. So you want to either publish them in a paper or present them at a conference and defend your work and why you did what you did and what you found. And this, as a scientist, is the most fun part. It's talking about your science with other people who are interested. Okay. So with that, we have finished this first part about the scientific method. And now you want to do the assignment and the quiz.